so my general interest, I just realized, so I come from turbulence, that's what I did before, and then I switched to biology for a long time, and now I realize that fluids are still a main theme of my research. The thing that mostly interests me is that biology evolved in a fluid environment, and so there's a number and increasing, amazing, enormous amount of numbers of adaptations. So biology knows how to read fluids, how to do things with fluids. And this is actually gonna be multiscale, so it's perfectly on theme. I'm gonna talk about fungi. I'll tell you why fungi are awesome, but just to illustrate what I mean by multiscale is these are spores. There are 8 spores. You know that fungi make spores. They are micron in scale. This is a fungus that ejects a lot of spores together. These are centimeters in scale. And also, spores travel across continents. So there are decades of spatial scales that are coupled. And I'm gonna give you like 3 little chapters that open into the adaptations that fungi developed on these 3 different scales. Okay, so, okay, this is working. So first of all, why fungi? And if you know something about fungi, you have probably seen images like this. They make the cover of Nature and Science, and people are scared because fungi kill animals. And there are entire species that are in danger. It looks like these are emerging diseases, so diseases that were not there before. So people are interested into fungi because of this reason. And these numbers are there because they are meant to scare you about fungi. Okay, this is, you know already, this is not the end of the story. There is much more to it, and in fact, most fungi are not pathogenic. They are symbionts, they are good for the environment, they are decomposers, and they do something that, that is essential for ecosystems. So here is one example, and it's very broadly available. So here is a pine. These are the roots of the pines, 1, 2, 3 branches. And here is how the pine actually lives in nature. So what this is a fungus. This yellowy network of hyphae, this is how they are called. This is a fungus. It's the body of the fungus. So when you think fungi, you probably think mushrooms. But the main part of a fungus is this network that grows underground, and it's typically good. Like in this case, it's a symbiont to the pine, and it goes scavenge for nitrogen in exchange for carbon. Fungi don't make carbon. They are not photosynthetic, so they need carbon. Okay, less obvious thing that I want to mention is because fungi are essential, we really should be able to make a list of a kind of list about what are the fungi that needs to be preserved. Okay, it's easy to identify a panda. We all really care about pandas, but we should really care about fungi too. And it's not obvious. It's not obvious how to identify them, how to decide which ones are important, which ones are in danger. All of this is really hard. And I'm gonna tell you something about how physics can help. A little bit, at least, hopefully. Okay, so before I get into the nitty-gritty of the talk, here is a fungal life cycle. And although there's a lot of names here that don't really matter a lot, there's one thing that you immediately see. There are 2 circles, one small circle and one large circle. And this, I want you to remember that there are 2 phases in a fungal life cycle. One phase is the phase where the fungus is happy. This is the small circle here. So there's food, the conditions are good, and the fungus is growing. So it gets nutrient from the environment. Remember, it's a network, gets nutrient from the environment, grows. It also produces asexual spores that bud off right there, and it keeps growing. There are fungi that are as large as Oregon. Okay, so this can keep going for a while. But then something happens, and then the fungus transitions to this big part, this big circle in this figure. This is the sexual part of the fungal life cycle. And this is triggered typically by stress. Okay, something wrong happens, I find a mate, I make sex, I make children, and then I let them go. Okay, this is the reason why fungi make spores. And it's the reason why spores are the motility mechanism for fungi. They don't have wings, they don't have legs, but they make spores and they let them go. Okay, so the goal of sexual recombination and spore dispersal is to explore a new environment. I want you to remember this. Sexual spores equal explore new environment. Okay, and this is the reason why I'm interested in spores. And here's is what I was telling you about. This is actually multiscale because spores are microns inside, but they can explore the entire globe. Okay? Is there any question until here?
I mean, in the small circle, they are happy they still make the spores.
They make asexual spores. That means there's no recombination. Okay, so it's a material, the genetic material comes from the fungus. So it can be complicated because the fungus can have nuclei that have different genomes because fungi are more complicated than us, but it comes from the same individual. Okay, the material for the asexual spore comes from the same individual. And these typically are not ejected to explore a new environment, but they just bud off. So it looks like it's just a way to keep growing right there. Yes?
What is the lifetime of the spore?
That's a very good question, but you're going to have to wait an hour. The bottom line is it can change between an hour and months. Okay, we're going to get there. So this is the outline, and I'm going to give you, so there's 3 glimpses at different adaptations that fungi put in place to cover these 3 different scales. So the micron scale story is a story about biomechanics. Okay? So you've seen this movie in the first slide. What this is these are 2 cells. They're called asci. The singular is ascus. I'm probably going to mention this word again, so you might want to remember it or write it down. Ascus is this cell, this long elongated cell. It's a very interesting cell. And in this class of fungi that are called ascomycetes, makes sense, this is the way it functions. These 8 discs are 8 spores, and the ascus, so these cells, swells. And when the spores are ready, there are osmolytes that come in, there are channels that open up, water comes in, this guy swells, and then it explodes. Okay? We're going to talk about this process for the next 15 minutes, maybe half an hour. Okay? So spores are ready, ascus is swollen, explosion, high initial velocity. How high?
What was the express spore on the top of the...
This guy is?
The top of the other one. The tip.
Oh, this one.
Yeah.
So the way we did this, these experiments is you take a fruit body, you squash it, so some spores get ejected, and then we randomly found one that, because I didn't say this, but the way we started this whole project was, okay, let's try to image this at high spatiotemporal resolution. Let's try to see how spores get out of the cells. Let's try to understand this biomechanics by looking at it. And the trouble is this happens between one frame and the next. So you see here, for a while, you have a second cell, and then there's no more cell, and so there's no information. They are there, and then they are away. So this gives us a bound for this species. Ejection velocity is how much do you think a spore is ejected at? How much do you think you reach with this osmotic pressure gun?
50 kilometers per hour.
I don't know in kilometers per hour, actually. Meters a second? Yeah, kind of, yes. That's pretty accurate, actually. It can vary a lot, but in this case, we have a bound because we know ejection happens between one frame and the next, and we know how long it is from one frame to the next, and we know how far they travel because at least they need to travel this distance. In this case, is at least 15 meters per second. So
it can kill you?
It cannot because it's too small first, and second because we'll see how far it goes.
But if you're an ant, it can kill you.
If you are an ant, that is a millimeter from. It's a good question, actually. I don't know, but I mean, one thing. So good question. It's a good question. So, how far does this thing go? Okay, so let's pass to the next slide. How far does it go depends on how fast it is ejected. And we've started to discuss this a little bit, but let's discuss it in a little bit more detail. Okay, so here in this frame, you only see potential energy. And in this frame, you only see kinetic energy. Okay, so if you make the idealistic, a very optimistic assumption that there is no dissipation in the system, then you know what's the final velocity because you equal this 2 and you know that the velocity of the spore is the pressure in the ascus divided by the density of the spore. Okay? So this, by the way, this is something that one could calculate what's the internal pressure in these pressure guns. This depends on the concentration of osmolytes. And if you use the diluted expression, the Van 't Hoff expression, you'll know that for 100 millimolar, which is what is typically measured for osmolytes in fungal spores, this is several atmospheres. Okay, so if you plug the numbers in here, you'll know that the velocity of the spore is of the order of meters per second. Okay? Okay, but this is very idealistic, as I said, because there's dissipation. And dissipation in the system occurs mainly because of the details of the tip of the ascus. Okay? The ascus, remember, is this elongated cell. Okay, at the tip of the ascus, there's some mechanism that prevents the spores to get out and then eventually lets them go out. Okay, and in a lot of species, this mechanism is simply a ring, an elastic ring. So the ring is closed, and it holds the entire pressure, these several atmospheres of pressure in the ascus, and then at some point something happens, it lets the spores go. And as they pass through, dissipation happens in the process. Okay, so this is the spore. This is a magnification of the apical ring. This is the way it's called. The spore is coming out. The ring is opening up a little bit as the spore squeezes out. What is the dissipation in this system? Okay, and I'm going to show you in the next slide that dissipation is dictated by the morphology of this cell. Okay, so somehow the way you design this ring determines how much dissipation there is in the system. And how many of you are physicists or mathematicians? Okay, how many are not then, which is easier? Okay, maybe I should ask you, what do you think the bottom line of this part is? I'm talking about this. I'm saying in an ideal situation there would be no dissipation, and now I'm talking about where the dissipation comes from. Where do you think I'm aiming at? Because I think most people will imagine that I'm aiming at saying fungi are great, they minimize dissipation. So I told you the answer, sorry, because I think it was unfair to pick. Okay, so the bottom line of the next slide, the next couple of slides will be fungi are awesome, they minimize dissipation, and it's not obvious how to do it. Okay, so let's look at how dissipation happens in the system. So the point of this slide is to convince you that there is a way to minimize dissipation. The reason is because look at how the spore is coming out of the ring. So this is the ring, it's a cross section, okay? So this is an elastic material. Here there's fluids. Here there is the spore, it's coming out. When, as the spore is coming out, there is a thin layer of liquid that covers the spore and that gets out with it. Okay, this thin layer of fluid, if this layer of fluid is very small, then viscous dissipation kills you. Okay, what is it? A viscous dissipation occurs when you have a fluid and you're trying to shear it. So you're trying to keep a layer of the fluid fixed and another layer very close to it sliding on top of it. Okay, this is what happens here. Why? Because fluids are at rest close to solid boundaries. This is not always the case, but maybe yes. Okay, so the fluid is at rest here at the ring surface, and it's moving with the velocity of the spore right here. So if this layer is tiny, the shear stress is huge. The gradient of the velocity is huge. Okay, so this is not good. Making it tending to zero is not good. On the other hand, if this is huge, you have a lot of fluid that comes out with it. Okay, and fluid has been observed to come out, drops of fluid come out with the spore actually. Okay, and if this happens during ejection, that's not good because the fluid is what keeps the cell under pressure. Okay, so you lose pressure head. Okay, so there is an optimum which is in between. I didn't tell you how much this optimum is, but if you balance these two energies, the energy loss due to viscous dissipation and the energy loss due to loss of pressure head, then you'll get what the optimal H is. H is the distance between the spore and the ring. Okay, it's the width of this fluid thickness, the fluid layer. Okay, great, yes?
What is uppercase L again? Picture?
L is the length of the spore. Okay, and it goes away because, you know, it doesn't matter. It's a little unintuitive, but if you have a longer spore, you have more dissipation, but it doesn't impact the optimal thickness because it's both energy losses are proportional to L. Okay. Okay, great. So is this the end of the story? Yes.
So what's the optimum when the energies are equal?
Well, if you sum two things that vary.
You have chocolate in the box.
If you have two things, one is 1 over, you can actually in this case demonstrate it analytically, but intuitively you see it because one is 1 over H, the other one is H. If you sum these two, you'll have this. Okay, sorry. If you sum these two, you'll have something that's like this. And this is the point where there is the minimum. One is 1 over H, the other is proportional to H, but you can do the derivative. Here it's everything is analytical. Okay, this is the E1, this is E2, and this is E1 plus E2. Okay? Yes?
So here the velocity of the spores coming out depends on the dissipation of the loss of energy.
Yeah.
So then, so we have U here, but we don't know what the U is because we don't know the loss of energy.
That's a good point. So if you put all of the numbers in, although I'm saying that dissipation is really something that we should care about, and and I'll tell you why I think that this is true, the absolute value of the final velocity that you get at this point, at the optimum, is not very different from the ideal case. Okay, so to leading order, you can ignore dissipation to get the order of magnitude for U and use that to plug that in here. But this is maybe that was the answer to the question, are we done? Yes, okay. So let's get back to the question that I posed. Yes?
Doesn't it depend on the volume of the spore? Because if it's very big, then the pressure will get lower when it gets out.
So that's a very good point. It's hidden in here, in fact. So there is a non-dimensional number that measures the volume of the ascus with respect to the volume of the spore, precisely for that reason. Because if it's huge, then whatever happens to the spore and the little fluid that comes out with it doesn't matter. So the magnitude, how stringent this condition is, depends on this non-dimensional number, precisely for that reason. Typically, so what I showed you in the movie, the volume of the spores, the total volume of the spore is typically much smaller than the volume of the ascus, but it's not orders of magnitude smaller. So that non-dimensional number, in fact, matters for the numbers, for the prefactors, and so we've done simulations for that. And it doesn't change the picture, the big picture. Yes?
Sorry. To get the term on E2 there, second dissipation term.
Yes.
Can you just, what's the main idea to calculate the energy loss?
It's essentially the volume that you lose. So W is the width of the spore, L is the length.
Just the volume, okay.
It's the volume of this fluid layer. Yeah, you're right. I didn't walk you through this. So this is H times L times W is the volume that is of fluid that's contained in here, that comes out, the order of magnitude. Here are, okay. And here, so just for completeness, this is the gradient, this is the viscous dissipation. It comes from the gradient of the velocity. So U divided by H times mu is the viscous stress. And then you multiply again by the surface, but because the stress is a pressure, so you need to multiply by a surface, and that surface is L times L. Okay, so back to the original question is, are we done? If we understand everything that's written on this slide, and if we all agree that this is correct to leading order, are we done? Is this the way that fungi can minimize dissipation in the system? By choosing this gap thickness? Is that right? Do you think it's reasonable from the point of view of a biological system to dictate what this gap thickness is? No opinion? Okay. I do not think that this is reasonable, because how is a fungus supposed to have a gene that controls this gap thickness? Gap thickness is the result of the dynamics, right? There is viscosity, there is a fluid here, there's elasticity here, there's a difference in pressure. The dynamics, so the physics, determines that gap thickness. It is pretty unreasonable to think that there is a gene that controls the gap thickness. There's actually no way you can do it. Or
evolution.
Nothing that, sorry?
Evolution can do it, no?
Well, evolution is going to be able to control morphologies, mechanical properties. How are you, so this, okay, so maybe let's do this. This is a magnification of the contact between the spore, which is now this huge gray thing on the left, and the ring, which is now this huge white thing. This is the cross-section. And this pink thing, this is the fluid that's in between them. Okay? What selects the thickness of this fluid layer is a combination of the fluid dynamics of this pink layer and the elasticity of this ring. Okay? So if you, I'm not sure that I'm going to be able to convince you about the scalings because it's quite complicated, but at least from the point of view of, with your hands, you understand that if I change slightly some properties of this fluid or some properties of this apical ring, or if the difference in pressure changes, then this fluid layer will have a different thickness. Okay, so the best I can do is to control my properties to select for this, for the dynamics to select for this optimal fluid layer. Okay, so if I was God and I knew what the fluid layer thickness was, I could, design a gene that controls the fluid layer thickness. But I don't think that this is a very reasonable assumption. I hope that everybody agrees. And so everything I can do is control the mechanical properties, the morphologies of the entire system, and try to make it the dynamics select for the optimal gap thickness. And I'm going to tell you a little bit more about it. There's going to be a lot of math. I'm going to try to walk you through it. It doesn't matter if you catch all of the little details of it. I want you to catch the idea. Okay, so first of all, the fluid layer is very thin. Okay, so this allows you to write this equation, which is an approximation of the full Navier-Stokes equations under the assumption that this fluid layer is thin. Okay, so thin with respect to its length. On the other side of the slide, you have an elastic ring. And if you ignore for a moment the presence of this fluid layer, then you can write a series of equations that tell you how the ring would be deformed by dry contact. This is called the Hertz solution. It's the solution of the the linear elasticity for a ring. Okay, and these are the scalings for this problem. I'm gonna walk you through the meaning of the symbols. H is the fluid layer thickness, which varies. H0 is the value that I care about because it's the order of magnitude of the gap thickness in the contact. But H varies, right? As soon as you leave the contact, this is x equals 0 is the point where the fluid enters into this thin gap. Away from x equals 0, you have that the gap thickness increases. Okay? So h, this is a variable, and it depends on psi, which is a complicated, where is it? Okay. Psi is a complicated length scales that depend on L, which is morphology, and dynamics. E divided by PR. E is the elasticity of the ring. So this is material parameter, gene for it maybe. PR is the pressure inside the contact. Okay? What is PR determined by? Well, to leading order, if there was no gap thickness, it would be just a static ring deformed by the presence of the spore. Okay? The spore is huge and it's squeezing out of the ring. So mainly that PR to leading order is determined by, again, static linear elasticity. You take a ring and a perfect ring, you deform it, okay? You impose a displacement, and this is what you get out of it, okay? This is where everything matters. E, the elasticity of the ring, W, the width of the spore, and all of these length scales that determine the morphology of the ring. Okay? I don't want you to understand this, but just to know where all of these complicated expressions come from. Okay? So then what you have to do is you have to look into the literature, because probably somebody has solved the problem for you. This is probably the most useful thing I'll say today. Read the literature before you do something. And you find out that in the 60s, there are people who have solved this elastohydrodynamic problem by realizing that major things happen around the point of entrance of the fluid into the contact. And then the rest, you can describe it easily by ignoring either elasticity or the fluid layer. Okay? And by this trick, with perturbation analysis, and then you can do numerical techniques that go back and forth from between before the contact and after the contact, you find out everything. You can do the scaling analysis of these equations around the contact, and what you get is 1, 2, 3 scaling relations for the 3 unknowns, which are h, then there is p, and then there is this lambda, okay, which is a new length scale. It's the length scale over which it matters. The interaction between elasticity and hydrodynamics matters. Okay? Again, you're not supposed to understand every detail, but just understand A, this is a complicated dynamics. B, highly nonlinear expressions come just from the dry contact problem, okay, where all of the dimensions of the ring and the spore and the material parameters matter. C, you can understand this by scaling analysis around the point of contact. Okay? Zack, now we are back to my original question. So we know how the optimal gap thickness looks like. We know how the actual H is. So we know what is the H that's selected by the dynamics. Now we equal them, and what we get is a constraint. Okay? What is the constraint? Well, these are the summaries of the 2 slides before. This is the optimal H, and this is the gap thickness. Inside these length scales, there are complicated nonlinear expressions of all of the morphology. And this is how, if you plug in everything, this is how the constraint looks like. Okay, again, this is complicated. It depends on morphology and material parameters. But in principle, one could imagine a gene for every single expression here. Okay, in principle. So, just to walk you through it, this is the width of the spore. This is a numerical constant that comes from the numerics. This is the combination of material parameters that we're going to assume to be constant across species for a moment. Okay, and we'll talk about this later. And this is the nonlinear combination of length scales of this ring. Again, it's a ring, so it has 3 dimensions: the length, the width of the initial ring, and the thickness of the ring. So these are D, L, and B. Okay? So now you take a good PhD student that has a little bit of time in his hands, and you look through the entire phylogeny to look for species that have this ring. Okay? So this is the tree of life. Here's fungi, here's us. Look, fungi are closer to animals than to plants, remember? These are the Ascomycota, all of the species that have this elongated cell and this squirt gun. This is the phylogeny of the Ascomycota, and these are the portions of the Ascomycota tree of life that have a ring, where there are species that have that ring. So then you go into the literature and you look for species in these clades. And Jörg mainly found 2 big clades, completely independent origin of this spore. And he looked for TEM sections. TEM is crucial because otherwise you cannot measure the morphology of the ring. You don't see it. It's like the movie I showed you. You don't see the ring. But if you were to do TEM, you would see the ring. Okay, so he looks for TEM sections, and he finds 43 species. Now, again, assuming that this is constant for a moment, this is a linear relation between W and this complicated nonlinear combination of all of the ring morphologies. And of course, I'm here, and so it kind of works. We were pretty pleased with this. Now, to decide whether you really are pleased or not, you have to decide whether this 2% is good. So what this tells us is that actual real morphologies of species look like they're optimized to minimize dissipation. Of course, 2 within a certain error. And you have to ask yourself whether 2% is satisfactory or not. Why do they care about this to this level of precision and not more or not less? Okay? And the answer is nobody knows, nobody ever knows, but this is consistent with different stages of this spore ejection mechanism. It's been proved that spores are streamlined so to minimize drag once they are in the air. And that work also shows that optimization is operated by evolution to within this limit. So this is kind of like, okay, so if I believe that work, I believe this work too. This is as much as we can say, actually, at this point. Okay, so this is the end of the microscopic part. I'm going to comment on this and then take questions. Okay, yeah, so we've done a lot of sanity checks to make sure that we are actually seeing something that's driven by evolution and not by random constraints of this complicated machinery. And this is the main one, so I'll just show this one. This is the line that I showed you in the previous slide. I just squeezed it so that I can fit all of the other points. So the red points are the optimal morphologies of the 43 species I told you about. And then these blue dots that appear here and that are much less optimized, actually these are not optimized at all, are additional species that have a ring, this ring at the tip of the ascus, but that is not functional. Okay, so there's no reason why evolution should keep the species onto the optimal line. You can do things with this data, like you can plot how far they are from optimality depending on the last ancestor that you know of that had a still functional ring, and it sort of correlates. You can do other things that I'm not going to show about, but the bottom line is this is consistent with the idea that evolution kept the species close to optimality, and it's not a random phylogenetic constraint or anything like that. Okay, so conclusion for the first part are I've showed you the biomechanics of a fungal cannon that uses osmotic pressure to squirt out spores at a very high velocity. And I've shown you that these fungal cannons are optimal in the sense of minimizing dissipation. This was done under the assumption that this number was a constant. And so I'll walk you through this. Mu is the viscosity, the dynamic viscosity of the fluid, the epiplasmic fluid inside the cell. Rho and P are the initial pressure at ejection, and rho is the density of the spore. Okay, these are to the power 1/2 and 1/4. The major component, the major term in this expression is P over E because it's to the 3rd power. Okay, what is P over E? Is the pressure at which the cannon ejects the spore divided by the elasticity. Okay, and our fit, we have one fitting parameter, which is this number. Our fit points to P over E, which is a fraction like about 0.2. So our results suggest that species broadly conserved this P over E. Okay, and one idea is that this P over E is 0.2 so that there is a safety criterion. You don't eject and you don't develop enormous pressure in the ascus that maybe the ascus is not able to sustain. Okay? Yes?
So you said that in the beginning the fungus has 2 life cycles, one under stress and one without stress in good conditions. This ejection only happened during this stressed part of
Yes.
the life. And so then my question would be, have you measured different of these material parameters of the fungi in different conditions? Because if the thing is due to morphology, then you would expect that that can never change. So the fungus might be regulating when to eject by changing viscosity of the fluid or something that's easy to do rather than the actual physical shape.
Okay, so the first answer is the mechanism that buds off the asexual spores, so the one in the small circle up there, is completely different. So there is no ascus that contains the spores. There is no ejection. They grow on the surface of particular structures, and then wind will make them break. Okay, so there is no. It's a completely separate process. They are not the same spores, not the same mechanism. Okay, so there's no mu or no p to be measured in that other case. The separate question is, how do you measure these numbers? And there are ways to measure these numbers for the sexual part, but they are very indirect, because you can imagine you have something tiny that is under pressure. Whatever you do to it, you're going to perturb it. But there are ways. There are to measure P, you can measure the osmolyte concentration. So you take a lot of them, you squeeze them, you take the fluid, you measure with plasmolysis. Biochemistry knows how to do this very well. It's been shown that it depends on the species. So one of the general idea is that each species will have a different way to develop this high pressure. Some will pump ions by opening channels. There are stretch-gated channels, so it looks like this could be coupled. But this brings me to the actual point of this slide, which is, if this is true, the question is, how do you regulate this P over E? And remember, something that I didn't tell you about but is very interesting is that these cells, these asci that contain the spores, there are no ribosomes. If you look for them, there's very few ribosomes. There's no active gene expression because the entire DNA is contained in the spores that are sealed. There's no communication between the spores and the fluid, and the epiplasmic fluid. So there's no transcription, no translation, yet this cell is able to elongate and eject the spores at the right moment, the one where P divided by E is 0.2 or about 0.2. Okay, so the question is, how does this happen? And the ideas we have is it's probably regulated by the biomechanics of the ascus wall. And there's not a lot known about it, but we are working on this. Yes?
I think you already answered, but my question was, how was it triggered? How this ejection was triggered? You were saying that it was like something breaking at the ring, or it's a completely different mechanism?
It's unknown. It is completely unknown. There's different ways that you could imagine it. There's actin, so there's this ring. At least in some species, actin is sort of surrounding it. So you could imagine that if you were to activate some myosin or some similar mechanism, you could have it open. But how does myosin or whatever mechanism correlate with this moment or with maturity of the spores? This is completely unknown. We have some ideas of how this could happen. That is one. The other thing is it might be chemistry. There might be something that sort of binds to, because some of these species have a little bit of a plug. It's not the pure ring that is closed. Some of them have a finite width, and there is a plug. So you could imagine some secretion of chemicals that. Okay, this is the end of the first part. So if you have more comments, yes.
So is there, if you have an ascus that has the wrong gap width, is there a correcting? Does it have a self-correcting mechanism, or is you get 10 different asci and you find that they miraculously have all the gap width is within 10% of the optimal?
you know, this is a very good question, and it's sort of like what we aimed at with that movie, but you've seen, you can't measure the gap thickness. That's not something you can measure. So the answer, I don't know. I don't know how, and you know, if you go back here, you'll see there's a lot of scatter. What does that mean? Does this mean that this is suboptimal really, or does it mean that they have a different value of the parameters? What does it mean? This is beyond the experimental possibilities.
But the theory that you have, it doesn't say for now whether it can self-correct, or it's not like finding a local minimum or something like this?
No, in the theory there is no local minimum. It's smooth. Everything is smooth. There is no stochastic term. Yeah, one could imagine theoretically to add noise and see whether there are local minima and see if the statistics would agree with that. But remember that these are 43 points over the entire phylogeny. So unless we find some other ways to probe this experimentally, I think it might be weak. Okay, so let me pass to the next part if that's okay, and which is going to be shorter. Okay, let me mention the next part and then jump to the third part because it's something that we are actually working on right now and I care about it. I care about the next part too, but I've talked about it for a while. Okay, so the next story is about centimeters and it's about cooperation. Okay, so this is something we could have done on the blackboard. I didn't think about it. Okay, if you have you ever worked on the ballistics of micro projectiles? Have you done this? No, right? Because who does it apart from people who care about spores? Right. So you probably think that meters per second is a lot, and this comes back to Alexandra's question. You probably think that meters per second is a lot, right? It's tiny, so it's going to go very far. Now, the trouble is when you are small, there is a term that you always forget about in the ballistics of macro projectile that kills you, okay? And that term is viscosity with the air, okay? And this is because your inertia scales with your volume, okay? But viscosity will scale with the surface, right? The interaction between you and the air happens through your surface. So when you're very small, your volume decreases much faster than your surface. Okay? And so if you're very small, you are killed by drag. Forget about gravity. Gravity is not in the picture. Okay? Your range will not going to be limited by the classic parabola of macroscopic projectiles. It's drag. Okay? And how do you quantify this? There is a time scale, which you might have heard about if you've done fluids. It's the Stokes time scale. It's this number. It depends on the radius of the spore squared, divided by beta, which is essentially the ratio between the density of air and the density of the spore. Okay? Nu, which is the kinematic viscosity of air. This is a number. Okay? And it turns out that for R in the order of magnitude of 10 microns, this tau is 2 milliseconds. Okay? The meaning of the Stokes time scale is how long does it take for a particle to react to the fluid that surrounds it. Okay? So say, for example, you're ejected at a huge velocity. It will take you a while to get adjusted to the fact that around you nobody's moving, the air is still. Okay? So that time scale is the time scale over which you decelerate to rest in our case. Okay? So in 2 milliseconds, the spore goes from meters per second to 0. How far does it travel? You can do this analytically, but order of magnitude is initial velocity times tau. Okay, so if you thought that meters per second were a lot, you might be sort of, you might change your mind. It's millimeters, right? If you are ejected at meters per second, you only travel millimeters ballistically. Okay, and now the question is millimeters enough? And this is a major question. Is it enough for what? Right? The only thing I told you about spore dispersal is that spores need to get out from the parent. This is the reason why you have sex if you are a fungus. Okay? 4 millimeters, is that enough? Well, if you look at the fluid dynamics of what happens around a solid object, you'll know that there is a layer of air that is still, okay? So that doesn't move. So even if you have wind right here, right next to the ground, there is a boundary layer, and that is still air. If the spore is ejected at these huge velocities and is not able to travel across that boundary layer, then we can safely say that this was useless, okay? Because it will just fall back from where it came from. And this is not the point of sex for fungi, okay? So, okay, this is what happens in a bunch of fungi, and this is how I got into studying fungi, because I thought this was really fascinating. This is a petri dish, so you know how big a petri dish is, like 10 centimeters. Well, maybe you don't know how big a petri dish is. It's about 10 centimeters. And here there are two fungi. These are mushrooms, so they are what emerges from the ground. So remember, underground there is this big network, and outside these fruit bodies, so the structure that holds the spore emerges. This is what we are used to think about when we think about fungi. These are the mushrooms. So these sexual structures emerge, and they all eject spores at the same time. These are hundreds of thousands of spores at the same time. Okay, so either we were wrong in saying that they only travel millimeters, or there's something to this synchronization. Okay, we will all agree that this is not just we probably made a mistake of a factor of 2 in the formula, or this is a factor of 20. Okay, so what's going on? Can somebody guess what's going on?
They push it on each other.
They push it on each other?
Push on each other, or there is some wind.
Either there is some wind. We're going to assume that there is no directional wind because it doesn't seem very plausible. But what do you mean push each other?
They are ejected like next to each other, and they kick each other.
They kick each other physically. There is collisions. This could be, but if you look at how far they are, the order of magnitude, and you'll see some more, it's not what's going on. So some of them go in a group. The most that you can see that goes in a group is 8. So it's true, this could be like a macro projectile. It's not what's happening in the species, and anyways, it wouldn't account for a factor of 20.
But these are spores, we're saying?
Yes, you're only seeing spores. There's no smoke, nothing to tag them. It's just spores. Yes.
Is it possible that because they're ejected so fast, they're creating their own air current?
That is good. Okay, let's elaborate on that. That is right. Okay, let's elaborate on that, because I did not think that this was possible for the numbers. You imagine there are tiny little spores. I mean, we know about the Tour de France, right? I mean, if you are in a group, you gain 20%. Who is a fan of biking? 30%? I don't remember. It's a fraction. It's not a factor of 20, right? So this is the first thing I did in fungal biology. So I'm emotional about it. So this is the trajectory of the spores. Okay, I is the index of the spore. It's transported by the fluid. So here is the velocity of the fluid, U. Fluid is the air, just to, okay. U is the velocity of the air. UI, big UI is the velocity of the spore. So it's transported by the velocity of the air. It adjusts to the velocity of the air in a time scale tau, which we already talked about. This is the Stokes time scale. UI is it. So if there was no velocity of the fluid, it would decelerate to rest in a time scale tau s. Now it adjusts to u instead of 0, okay, in the time scale to s. Then there is gravity, and this is the position of the spore, velocity, position. Okay, these are the Navier-Stokes equations. Okay, so the equations that, that dictate how the air moves. And we're going to assume that at time 0, there's no forcing, so the air is completely still. The only thing that will inject into the simulation will be the initial velocity of the spores. Okay? So how does that make the fluid move? This is the suggestion from the second row. It's the interaction between the spores and the fluid. Okay? The spores are decelerated by the fluid. The fluid is accelerated by the spores. Okay? This F, sorry, red is not a good choice. This F is the force that the spores exert on the fluid. It's action-reaction. Okay, the simplest possible model you can imagine. Yes?
Can you elaborate a little bit on the energy balance here? Because, you know, if some spores have helped, then the other spores must lose some energy.
I'm going to comment on this in a second. Okay, so first let's look at the simulation. So I told you all of the ingredients. The spores, there's I think 100,000 spores on this circle. The air is at rest. The initial velocity is a few meters per second. Now I don't remember anymore. This is the ballistic range. Okay? This is just to remind you what is the mechanism that we are trying to find out about. And here it is. So you see that little by little, there is a large-scale jet that develops purely due to this term, this interaction between these two. It takes a little bit to develop, which goes back to the last question. The first spores that are ejected, if you look at them a little bit, okay. If you look at the first spores that are ejected, you'll see that they are not traveling as far as the others. Okay? This is because it's unsteady. The moment it reaches its steady state, then everybody goes at the same distance. But there is a time scale, which is a macroscopic time scale, right? It's the time scale for this large-scale structure to develop. And along that time scale, all of the spores that are ejected are sacrificing for the collective, okay? So before commenting on this, I'm gonna tell you a little bit more about this jet. So what is the range of this jet? Remember, we started from spores that individually are killed by drag, okay? Now I'm gonna show you that the height of this jet is mainly limited by gravity. Okay, so there are two different regions of this jet. There is a first region where spores are clearly much faster than the air, at least at the beginning, right? The air is still, the spores are ejected at meters per second. Okay, this layer is very thin because what happens in a time scale? In a Stokes time scale, always the same time scale, the spores will adjust to the velocity of the air. Okay? The little twist is the velocity of the air will not be 0 at the end of this layer. It will just be given by momentum balance. Okay? You inject momentum at z equals 0. Everybody adjusts to the same velocity, big U, with no indices, which is just driven by momentum balance. Okay, you are accelerating a lot of material, so it's going to be much smaller than the initial velocity of the spore, but still non-zero. Okay, from now on, everybody's moving at the same speed, so that means this huge part of the jet is passive. Passive in the sense of you have a heavy jet that has an initial velocity with a given area, which we are going to assume is not very different from the area where the spores started, and this develops passively. Now this is the gravity limited height, okay? Just again, you compare forces, and you just determine how far you can go if there's no friction, if there's no viscosity. And it turns out that because gravity is more important than viscosity for these numbers we have, you essentially get very close to this maximum height. Okay? Viscosity plays a minor role. So the transition between this brown region and this red region brings you from a microscopic projectile that's killed by drag to a macroscopic jet that's mainly limited by gravity. You can't do better than that. Okay, so this is the point I wanted to make, and just two quick comments. The first comment is we've done PIV to demonstrate that this is right, and what this is showing qualitatively is that, so again, this is the fungus. These are individual spores that appear as streaks, and here are spores that appear as points. If you have a fast camera, you can infer what the initial velocity was. Well, here, well, whatever. This is not working anymore. You can infer the initial velocity there just by dividing the length of the streaks by the exposure time, and then the points just tell you that they're not moving enough in an exposure time. So this is qualitatively demonstrates the point, and you can be more quantitative. You can do PIV, you can measure the entire profile, the velocity of the center line, how long it takes for that to decrease to zero, and it works pretty well with the theory. Okay, I'm not gonna comment on this because I want to pass on the next part. The only point I want to make is that so there is a major underlying question, which is cooperation here. And we've talked about conflicts that might arise from the fact that the first spores contribute to the cooperation but don't get as much as the last spores, because it takes a while for this cooperation to prove fruitful, right? So initially we thought, and this might be true for some species sometimes, there are waves that coordinate the spores across this fruit body. So if you look from above, this is what these two movies are showing you. If you look from above, and the black dots are the spores, if you see a white dot, that means that the spore was ejected. And you see sometimes this happens across in a wave. And this is a way to also not only allow them to communicate, but also to enforce cooperation. Sometimes the wave is not there. So this is something that we're working on. What drives this wave or this communication? We think it's mechanics, because you see the fruit body sort of shrinking when more and more spores are ejected. We're working on this. Just an idea to throw there and to conclude. Okay. So this is the conclusion that I already gave you for this part. I think if you have like a couple of questions or comments, we can go through them, and if not, we pass to the next part, which is more, yes.
Is it fair to think, so when you set up this problem, you talked about the mean estimate, right? So you said the time scale and so on, and then on average they will travel only a couple of millimeters. But if your velocities develop a distribution, and you look at the tail of the distribution, then of course, you know, they will travel far in the tail. So it's
so I think this brings me to the next part. I'm sorry for you if you had, which is uncertainty. Okay, let me first give you the speech, and then I'll answer the question. Okay. So I've told you two stories at two different scales where it looks like fungi have everything under control. They're great, they know how to optimize things, they also cooperate to get farther. But the trouble is, after the spore leaves the fungus, and after this few centimeters where there is maybe, for some species, this macroscopic structure, then the spore is a passive object in the atmosphere. And if you know anything about weather forecasting or turbulence or anything related to how things travel in the atmosphere, you'll know it's dramatically stochastic. Okay? So how do you make the connection between something that looks like the spore, the fungi care about, and something that looks like fungi will have no control about? It's all part of the same process. Okay? And it's kind of a little bit like the question that was asked. Okay, so specifically we're going to focus on one observation, okay, that's depicted here. And it is about spore liberation. When do fungi decide to liberate their spores? These are two examples, okay? There's plenty of this in the literature, where the concentration of spores of a given species is measured at close to the ground, but close to the ground means here. So we're going to assume that this is a proxy on when spores are liberated, okay? And the data look quite different from each other. There are species that look like, so this is the concentration of spores of that given species over 15 days. And if you count the peaks up there, there'll be about 15 peaks. So it looks like those species discharge their spores reproducibly every day. And then there are some other species that don't look like they're that regular at all. Okay, and where does this difference come from? One more piece of information. If you focus on those species up there and you look at the times of the day when the spores are ejected, again, it looks like there's a lot of variety. There are some species who prefer to eject during the day, like those up there. Okay? And some other species prefer to shoot at night. Okay? So it looks like there are preferences over which spores are liberated. Does this have anything to do with the fate of the spores? Okay, what happens to them in the atmosphere? Okay. Okay. We're going to take the point of view of time and not of space. So I've told you about ranges a lot. Now, for what happens in the atmosphere, I think that the major point that we should care about is time. How long does it take for spores to travel from A to B? Okay, not too much how far it is, because I told you we have no way to determine whether 5 meters versus 5 kilometers is good or bad. I have no idea how to do that. The only thing I know is it needs to travel across the boundary layer. But then after that, I don't know how to pose any constraint on what happens at larger scales. Time is different, and it's different because of this reason. These are data, and again, there are more data in the literature, which shows you the lifetime. Again, this was a question that came from about here, but maybe he's not. Someone here asked how long do spores live. Okay, this species only lives an hour in plain daylight. Okay, this is the comparison of the fraction of spores that was alive after exposing them to light. The left one is a control, no light. The right one is the experiment done trying to reproduce the daylight in the environment where the spores were isolated. Okay, so it looks like these spores only live an hour in the daylight. These are the champions that I found. It's probably not the best, but some species were found at 41 kilometers from ground, so well high in the stratosphere, not even in the atmospheric boundary layer, alive. Okay, there are thoughts about a microbiome happening in the troposphere and in the stratosphere, including spores that would metabolize things high up in the atmosphere that would survive for weeks. Okay? This is the other one that I found. This guy only lives 0.7 hours in the full daylight. Okay, so this is the reason why I think time is different. Clearly, there are species that develop long-lived spores and species that did not. Okay, so this is one thing that we can compute. How long does it take for species to travel from A to B in the atmosphere? Does this depend on the strategies for spore liberation, which is something that the fungus can control? Okay. So I only have preliminary results for this, so I don't have a definite answer, but I have some answers. So this is our twist. We're gonna think about time. Okay. So this is, again, you take a PhD student that then becomes a postdoc, and he's talented, and he knows how to do simulations in the atmosphere using meteorological data, which is needed here. So this is the equation of evolution of the trajectory of the spore, and you see it evolves because of three terms. There is U-meteo. These are gigantic data sets of meteorological winds. Okay, these are becoming increasingly resolved. They are large scales, so the grid points over which you know the wind are like maybe 10 kilometers apart, 1 kilometer apart from the best resolved data sets, and 3 hours apart. Okay, so this is how the large-scale landscape looks like. Then you have to add an important term, which is the turbulent fluctuations, and you're going to have to model this because there is no measure that tells you exactly what the turbulent fluctuation at that point in that instant in time was. And then there is sedimentation, okay, which means gravitational settling. So these are heavy particles. Tau is Stokes time scale, okay, always the same, and that's the gravitational settling, okay? So then what Daniele did was he took 10 points across North America, okay? We decided to try and be more detailed in the times rather than in space. So we took just 10 points because this is already a lot of simulations, and we tried to make them spread apart, trying to explore different niches so that our results wouldn't depend on like being close to the sea, to the mountains, to the lake, some specific microclimatic pattern and stuff. And then he took, he released spores at a certain height, which doesn't really matter that much, for 8 simulations per day. So to explore how the trajectories depend on time of the day. And then he tried to understand the statistics of the flight times, how long it takes for spores to sediment. Okay? He's done the simulations, so as I said, from 10 locations, 8 simulations per day over 4 months for 10 to the 5 spores per simulation. So this is a lot of data, and now we'll try to understand them. Okay. So this, just to give you a glimpse, is the PDF of the average flight time. So you take one simulation, you take the average time it takes for spores to sediment, and you see it's very spread out. Yes?
How do you measure when it is sedimented with the spores?
How, sorry?
How you know the time when it is sedimented?
Well, you are following the trajectory of all of the spores. Yeah. You see it starts from a certain height. When it arrives at 0, that's the time. You'll stop integrating the spore. You'll say, okay, destination.
And some of them you don't.
Some of them, this is a good point that I didn't put data for it. Some of them will go into the stratosphere, and then the simulation, because the simulation has he did like 6 weeks. He's not gonna simulate forever and ever. Also because the spores that pass into the stratosphere then are taken by huge synoptic winds and get out of this volume of integration. So that's another point that we are quantifying. So the statistics you have to know will be biased onto those spores that ended up on the ground within the 6 weeks. Okay, so it's very spread out. There are spores that take only a few hours and spores that remain in the atmosphere for weeks. Okay, one thing that we realized is that for this question, this specific question of how long it takes to come back, only the vertical direction matters, right? We don't care where the spore ends up. I only care about when it comes back to ground. So for this specific question, it doesn't really matter a lot what the wind is, which is Great, because it means we can do much more simulations. We can just simulate models of turbulence, because that's all that matters. It's not great for Daniele, who did all of the simulations, but I think people are going to appreciate that there are some questions that don't need those enormous simulations. The other thing that surprisingly doesn't matter that much is sedimentation. Okay? Sedimentation only matters where turbulence is very weak. So either very close to the ground, so it matters to model the interaction, the deposition, the interaction of the spore with the surface, or in situations where there is no turbulence at all. Okay, that I'm going to show you is what happens at night. Okay, so if you look in the literature, I think a lot of work has been done on those two terms, but we're going to focus on that one term that's interesting for us to understand the flight times. Okay, specifically, and this is all I want to show you. We're going to look at fitness, which in this case is very naturally defined as the fraction of spores that sediment before their lifetime. Okay? For the moment, let's assume that all the spores are identical, so no variation. Every spore lives, in this case, I think I took 6 hours. Okay? It can be done for any of these lifetimes, and which is interesting. I'm just going to show you 6 hours. Okay, this is how the fitness depends on time of the day. Okay, what this means is that if you throw your spores at noon, they are gonna take a while to come back. Okay, and this is because at noon there's a lot of convection, the atmosphere is unstable, so they're gonna be taken high up in the atmosphere, and it's gonna be a while before they come back. Okay, so good. So there is a dependence on time of the day, so those patterns might have a meaning. This is how to quantify this, and it's what I just told you about. On the y-axis, you have W star, so the magnitude of vertical turbulence, okay, as a function of time of the day for 30 days. This is January, and it's in Mexico. Okay, this is how it looks like. It's always high during the day, it's always low during the night. Okay, this alternation is called the diurnal cycle, and it's what causes that pattern. Okay, so all right, time of the day matters. Okay, it correlates with the time it takes for spores to come down. But if you only have a clock to decide when you're gonna discharge the spores, well, the error bar is pretty big, right? So is there anything better that we can do? This is the question. And there is probably something better we can do, because if we look into the details of that plot, of the fitness over time of the day, we'll look at that, we'll see that in different geographic point at different times of the year, the situation changes very much. This is Mexico in January, and this is how turbulence alternates pretty regularly between day and night, and that is the fitness over time of the day. Let's look at another simulation. This is Canada, the same month, and there's no alternation between time, between high and low turbulence. There's no diurnal cycle, almost nothing. Correspondingly, your watch is completely useless. The fitness does not depend on time of the day at all. And sorry, yellow is really a bad choice, but can you see the curve? It's just, it doesn't depend on time of the day. Okay, if you're in Canada in January, forget about your watch. Okay, and so now we are at the point where we are trying to quantify this property. And you can quantify it, for example, by looking at the autocorrelation of anything that relates to turbulence. Here I'm showing W star, but there's different parameters, like the stability parameters, length scales that tell you how intense is turbulence. You can do it for anything that is related to turbulence. You look at the autocorrelation and you see that if this was a sinus, it would go regularly from 1 to -1 to 1 -1 every day. So if you take the autocorrelation at 24 hours, that tells you how much of a reliable cycle is there. Okay? for turbulence. And you can do this for fitness as well. Okay? And what it turns out is, of course, if you have a reliable cycle, your watch is very useful. If R is equal 1, it means during the day and during the night, you know what turbulence will look like. And as a consequence, the reproducibility of your chi is the fitness, becomes close to 1. You almost know exactly how long the spores will take to come back down. Okay, it's not exactly for each spore. Remember, these are averages, there are statistics. But if you are on this part, if you're in Canada in January, forget about your watch. You have to do something else if you want to bias your spores to a good, successful life. So what we're working on right now is exactly this. What do you have to add in order to make better choices about spore liberation? And we have done something, but it's A, there's no time, and B, it's not to the point where I could tell you what the answer is. The only thing I know is that sensation will be needed mostly where cycle is unreliable. Okay? And we know where that is. We have maps now. Okay? And so with this, I want to conclude thanking you, thanking the people that have contributed to this, and the funding. Thank you very much.